She generated so many wonderful trainees throughout her career, and they are all spread out through the country. And so that is perhaps the biggest legacy that she will leave behind at some point. And the second, that she's really avid biker. So I'm sure that even in snow and rain, she's biking. And she negotiated a few trucks and cars and vans and all of that. She won every single time. So Connie, we're so delighted to have you today. And introduction will be made by this beautiful man, Jennings Liu.
Good morning, everybody. My name is Jennings Liu, and I'm an MD PhD graduate fellow from Dr. Palchevsky's lab. Today, it's my distinct pleasure to introduce our keynote speaker, the Bullard Professor of Genetics and Neuroscience at Harvard Medical School, Professor of Ophthalmology at the Massachusetts Eye and Ear Infirmary, and investigator of the Howard Hughes Medical Institute. Originally from Pittsburgh, Pennsylvania, she attended college at the University of Maryland, where she obtained her bachelor's degree with a double major in biochemistry and microbiology. She went on to complete her graduate studies at the Massachusetts Institute of Technology, earning her PhD and completing her dissertation research under the mentorship of Dr. Philip Sharp, who made the Nobel Prize-winning discovery that eukaryotic genes contain long sections of non-coding DNA, now known as introns, which are removed by RNA splicing to form mature messenger RNA. She remained at MIT as a postdoctoral fellow in the laboratory of Dr. Richard Mulligan, where she contributed to the development of retroviral vectors for the study of gene transduction and developmental biology of the retina. Over the course of her career, she has been the recipient of numerous prestigious honors, including the Kogan Award at Arbo, the Pressler Prize in Vision Science, and the Lisman Vision Science Award at Brandeis University. She was elected to the National Academy of Sciences in 2002, and also currently serves on the editorial boards of the Proceedings of the National Academy of Sciences and eLife. Her research is focused on development and diseases of the central nervous system, with emphasis on the retina and molecular mechanisms of cell fate determination. For instance, her laboratory is studying mechanisms of photoreceptor cell death in human blinding diseases such as retinitis pigmentosa and macular degeneration. More recently, their interest has been in developing gene therapies using AAVs to preserve vision in mouse models of inherited retinal degeneration by targeting genes that fight oxidative stress, inflammation, and/or provide metabolic support. Today, we have the great privilege of hearing about some of this latest work in her talk titled Gene Therapy Approaches to Prolong Cone Survival. So without further ado, ladies and gentlemen, I present to you Dr. Connie Cepko.
Well, thank you very much for all of those nice comments, Chris and Julian. I really appreciate it. I'd like to start with this initial image, which is one that was reconstructed of a mouse cone photoreceptor by Claudio Punzo when he was in the lab. And what it shows is the outer segment structure out here, inner segment, and the red staining is phospho-mTOR, and then the cell body here with a very thin shell of cytoplasm. So this is obviously a very highly developed specific structure. And we all know that this outer segment is something we need to preserve to enable the cell to detect light and carry out phototransduction. This phospho-mTOR is something I'll speak about later. This is here in the inner segment of these cones. So our goal is to preserve as much structure and function of cones throughout the process of several different diseases, one of which we focused on is retinitis pigmentosa. First, I'll tell you who's done all the work. So some of the work I'll tell you about is by Sean Wang, who was a medical student in the lab and then spent a year as a postdoc and now is at Stanford as a resident. Sawyer Shu, who's currently a postdoc in the lab. Ryoji Yamamoto, also currently a postdoc in the lab. David Wu and Wenjin Zhang were former postdocs who, unfortunately, I don't have a good picture of right now, but I'll be pointing out to you the various types of work that they've done. So this is a sort of simplified depiction of the various diseases that lead to loss of vision and some of the genes that cause those phenotypes. Retinitis pigmentosa, you can see here, has quite a few different genes. These genes perform all kinds of different functions. We have transcription factors, phototransduction proteins, structural genes that carry out structural roles. Some of these genes actually are shared with some of the other depictions here. So we don't really understand how we end up with all these different phenotypes from these different genes, but there's at least some commonalities that allow these various diseases to be defined. Retinitis pigmentosa, I think most of you know, is a disease where a person is born night blind or nearly night blind, usually due to intrinsic problems in the rods. So genes that are expressed only in rods, for example, rhodopsin, will lead to retinitis pigmentosa. And people who inherit those types of disease mutations start out with very good cone vision, that is high acuity color daylight vision. But the tragedy of this disease is that a person then loses their color vision. And this is a bystander effect because these cones do not express the disease gene. Similarly, the retinal pigment epithelial cells also suffer from bystander effects. Again, they don't express the disease gene. They also tend to fail in retinitis pigmentosa. So among these different diseases, we chose to study retinitis pigmentosa initially as a way to understand how cones die as bystanders and to come up with ways to combat that. Our hope was that there would be a common problem, since no matter what the rod disease gene is, the phenotype tends to go the same way. That is, as the rods die, then the cones start to suffer. But our hope is that anything we learn about keeping cones alive in retinitis pigmentosa may be relevant for some of these other genes, or these other diseases as well. I'll also say the mouse models for retinitis pigmentosa are pretty good. Some of the same disease genes that affect humans are also in some of these mouse models. So I can put a sort of a simple cartoon together for what I just told you, that in retinitis pigmentosa, there is a period of loss of function of rods. They typically die, and they do so at different rates depending on the disease gene, both in mice as well as in humans. And then there's a period of cone dysfunction and cone death. We actually charted this quantitatively in 4 different mouse models. And what we found is that the cones start to suffer when almost all of the rods in their neighborhood, their immediate neighborhood, are gone. And so this sets up some of our thinking about what the problems are that lead to the cone death. Now, we and others have come up then with suggestions about what might be causing the cone death. One possibility is that there are trophic factors produced by rods. Obviously, when the rods die, those trophic factors will go down. And as you know, Jose Sahel has been tracking the effects of RDCVF, such a factor that may be able to keep the cones alive, independent of the rod disease gene. Others have suggested that toxins are being released by dying rods. I think the kinetics shown here, at least in the mice that we've studied, rules that out. You would expect there to be a maximal release of toxins here at the peak of rod death. But there's really quite a delay before we see the cone death even beginning, and it can go over a very protracted period of time after all of the rods in the neighborhood are gone. So at least in the simple form, I don't think it's release of toxins from dying rods. Now in the literature and in some of our own work, we think that there's oxidative stress, metabolic problems, and inflammation. And we've set out then to address these different problems using gene therapy with AAV to directly combat some of these problems. So overall, then, our goal is to get in at this time. Now, you could imagine that we could try to treat before there's any cone death, or even save the rods with some type of generic therapy. And I think that would be great, but we haven't come up with any great ways to do that. So we're looking at the period then at the onset of cone death as a time point to intervene, eventually in humans. In mice, we go in at different time points, mostly pretty early because of some aspects of the technology. So I told you we measured the kinetics of rod and cone death in 4 strains. These are the 4 strains. We were lucky to get these from various colleagues. And they have different rates of death. So this one, rhodopsin knockout, is particularly slow. The phosphodiesterase beta, there's 2 forms, one very rapid, the RD1, and one a bit slower, the RD10. So what we did after characterizing the rod and cone death kinetics is we did an RNA study to ask, is there something in common across these 4 mouse models at the onset of problems for the cones? We knew there would be many RNA changes as rods die, but what is it that's inciting these problems in the cones? And by looking across 4 models,
I think we don't hear the speaker.
Yeah, someone just muted me. I got a little.
You have to be
message
careful,
announcing.
Connie, just to explain. I think there was noise coming from somebody, so I think the moderator muted everybody.
I got it. Okay.
Whoever's controlling it, when you mute everybody, please unmute Dr. Cepko. Thank
All
you.
right, I'll know now when I see that message what to do.
Okay, thank you.
Okay, so at any rate, what I was saying is we did an RNA study looking at what are the common RNA changes at the onset of cone death across the 4 models. Again, they have different kinetics and different rod disease genes. And we found there was a common signature. Most of the commonality was here in the metabolic pathways and the insulin mTOR pathway. Now, we hadn't really been thinking about metabolism. We understood that there would be problems as cells die, but we weren't sure what those would be or whether they would be causal. But we decided to start looking at this. And one of the first things we looked at was mTOR. Now, I'm sure most of you know that mTOR is a central regulator of metabolism. It's responding to various types of influences. For example, growth factor receptors can lead to its activation. When it's activated, it has a phospho group. When it's not phosphorylated, it's actually a negative regulator of growth. And you can see here some of the pathways that it regulates. So when it's active, it's basically giving a green light to growth. When it's inactive, it's a red light to growth. And again, it's responding to growth factor signaling, amino acid or nutrient levels, ATP, AMP levels. When it's inactive, it also can lead to the upregulation of HIF-1 alpha, a transcription factor that can lead to the upregulation of GLUT1, the glucose transporter, as well as glycolytic enzymes. So it's a central regulator of metabolism and overall growth, as well as macroautophagy. So we looked at phospho-mTOR initially in wild type. So this is the kind of depiction I'll be showing you. This is a mouse retina that's been flat mounted and now stained with an antibody specific for the phosphorylated or active form of mTOR. It's a bit of a peculiar pattern, I will say. We expected all cells to have phospho-mTOR in a healthy retina, but we only see dorsal cones. And that's what's shown here at higher mag in these insets. So you see it's actually in the inner segment. And the very first slide I showed you in this presentation showed you that nice red stain in the inner segment. So we're not sure why just dorsal cones and why not all the other cell types or all the cones, but it gives us an assay point to ask about the health of cones. So we then looked at the mutants, the degenerations, the RP models in mice, and we found that the first sign of trouble for cones was lack of mTOR phosphorylation. So here's a wild type on day 15 or postnatal P15, very early. If we look at the RD1, the rapid mutant, by day 15, when some of the rods are still hanging around, but a lot of them have already died, we've lost the phosphorylation. This was true across all 4 mouse models. And if we look even later, day 30, you can see now the cones, which are in green, are fairly misshapen, and they've lost their mTOR phosphorylation. So this is an indication that something has gone awry in terms of the activation of mTOR. We don't know exactly what that is. I'll tell you our thoughts about it, though. We wondered then if mTOR is not phosphorylated, is macroautophagy being unleashed? Sometimes cells will digest whole organelles like mitochondria with macroautophagy. So we looked for evidence of that, and we did not see that. There's another type of turnover of proteins that cells that are stressed can carry out called chaperone-mediated autophagy. And you can think of this as kind of a slow burn of selected proteins to keep a cell alive. And one can find evidence of that by looking for certain markers that are upregulated in lysosomes for cells that are undergoing chaperone-mediated autophagy. And we saw that in the cones and the RD mutants. So what you're looking at here is a flat mount, and we're using confocal microscopy to look at a green marker, LAMP2, for lysosomes. Again, this goes up during chaperone-mediated autophagy. Wild type cones, the blue is the level now of the cone cell bodies. We don't see this. In the phosphodiesterase beta or RD1 mutant, we see it. And in the same mutant, but going down one layer to look at the bipolars, we don't see it. So this is consistent with the cones being stressed out, having some type of perhaps nutrient deficit that leads to their triggering of the chaperone-mediated autophagy. We looked at a few other things to ask what might be triggering this lack of mTOR phosphorylation. Culturing explants, then, we were able to watch mTOR phosphorylation in wild types with and without glucose and other factors that might trigger mTOR phosphorylation. And it seemed the thing that regulated the mTOR phosphorylation in those explants was glucose. The presence or absence of glucose led to the lack of or the presence of the mTOR phosphorylation. So going back then and looking at the cones and the disease mutant, we also looked for HIF1 alpha upregulation and GLUT1, and we found that they were also upregulated. And now these things are all correlations, and we were trying to understand, is mTOR actually regulating, or its status, its activation or lack thereof, is it somehow impacting cone health, cone survival? So we decided to inject insulin to try to stimulate mTOR phosphorylation to see if we could then prolong the life of the cones and thereby get a little bit more sense of whether there was causality. So Claudio Punzo, who did all of this work, then injected the RD1 mutant with no, with no treatment, and blue indicates where the cones are. And I should say, in these mice, we get a central to peripheral gradient of cone loss. So at this stage, in this untreated mutant, we've got peripheral cone survival, but the central cones are more or less gone. 3 weeks of insulin injection, you can see we've rescued quite a few cones. And if we wipe out insulin production by wiping out the pancreas with a drug, we get even less cone survival. So this is a consistency argument. It doesn't prove that this is working through mTOR phosphorylation. It doesn't even prove that it's through cone, the autonomous effect on cones. But Claudio Punzo then went on in his own lab to genetically alter the level of phospho-mTOR in cones specifically. And he found that he could, by toggling the mTOR phosphorylation level, prolong cone survival. So there is an autonomous effect of mTOR phosphorylation on cone survival, and you can affect that with insulin. So if we go back then to our little cartoon and think about where does this leave us then, we sort of gave ourselves a green light to think about metabolism and perhaps nutrient shortage, glucose specifically, might be part of the problem for cones. But I also put up a list, and I'll go back to that list, of other problems that we know are also happening. Again, from the literature, this has been seen by other people in human tissue as well as other animal models. And we decided to just systematically go through some of these things and see if we could address these problems using, again, AAV gene therapy. So our overall strategy is to take AAV, have at least one of the vectors encode a test gene. We also put in a vector that has a cone-specific promoter, originally Jeremy Nathan's red opsin promoter. And we have that drive a nuclear form of GFP. What this does is it gives us a really quick and easy way to quantify remaining cones. We get a very high rate of co-infection. So we've monitored, if we put in, for example, a red and a green vector, we've asked how many of the cones are yellow, and we get about 95%. So we're able, at the doses we put these in and the time point, so this is P0, the day of birth of the animal, we can get really high co-infection rates, which allows us to test multiple vectors at the same time. Now we go at P0 in the RD1 mutant because we need to get the genes on board before the cones have died. And this is a very rapid degeneration, and it takes about 3 weeks for the AAVs to max out their level of expression. So going in early allows us to get those genes on board and hopefully see a survival effect. The second thing is, if we go in at P0, this is before there's a really good connection between the RPE and the outer segments. In fact, there aren't any outer segments at P0. So the inoculum flows through the entire eye. So we get the entire eye infected with, again, the GFP vector plus the test vectors. We also usually see a repair of any damage we've done with our needles. So it's a good way to look at an intact eye that's fully infected with the vectors prior to degeneration. We are also doing work to then deliver these vectors at later time points where it might be more relevant for human gene therapy. So what we do after these injections is we take out the retina, we make a flat mount, we quantify the GFP cones. We usually look in the center 50% because at the time points we're looking, that's where the cones have already died in the controls. And we assay visual acuity using the optomotor test. Okay, so first we looked at oxidative stress. Many of you know that oxidative stress leads to problems for proteins, nucleic acids, and lipids. You can see evidence biochemically of all of those types of molecules being oxidized. And there's a couple transcription factors that are pretty good at turning on many of the genes which fight oxidative stress. One's called NRF2 and one's called PGC1 alpha. There are also enzymes that directly detox free radicals, for example, SOD2 and catalase. So first we tried SOD2 and catalase, and I'll show you that data. And then we tried these transcription factors because we thought they might give a broader type of control of oxidative stress. And I'll show you what the data look like. So this is a control retina. This is an RD1 retina treated with just a control vector with GFP. And again, the periphery has surviving cones at this stage. and a lot of the central cones have died by the stage we're assaying. And if we put in one of our antioxidation factors, we get a lot more, especially central cone survival. And so we quantify this over time. So this is the age of the animal. This is again the RD1, and this is the cones in a particular area. If we go with just GFP, you can see we're losing cones rapidly through this time period. If we put in PGC1 alpha, you can see it's deleterious to cones. We don't know why. If we put in SOD2 and catalase, we get good survival effect at postnatal 50, but it wears off by postnatal 70. If we put in this transcription factor, NRF2, we get better survival at all the ages. It's more durable. And if we look at the oculomotor activity, I'll call your attention to this panel. With the NRF2 injected eyes, this is acuity on this axis, and this is age. So GFP injected or control uninjected, you can see rapidly lose their acuity at this time, but the NRF2 can preserve that acuity for a while. So NRF2 then is a very nice vector to use, and we then tested it in RD1, RD10 in the rhodopsin null, and we were able to see these types of survival effects and in acuity retention in all of them. So this is something then that we're thinking could be a generic solution to many different types of disease genes that are expressed in rods, or perhaps even in cones for other diseases, because the pattern of genes that it turns on is fairly broad. So again, antioxidative damage, but also it's anti-inflammatory, and it also affects some metabolic pathways. So then thinking about what other diseases might we be able to treat with NRF2, we thought about macular degeneration. I'm sure all of this audience knows about this, so I'm not really going to go through it. But most of you probably also know that we don't have a good treatment for the dry AMD. We do with the anti-VEGF for the wet AMD. And one of the models about what initiates AMD is oxidative damage and inflammation, particularly focused on the RPE cells. Perhaps complement deposition and other types of insults that hit the RPE lead to misfunction and then death of RPE cells. So we thought, could we look at an effect of NRF2 on RPE cells? And I'm sure most of you know that AMD is not well modeled, really, in most of the animals we work on. First of all, they don't have maculas, and there's just various problems trying to recapitulate the variety of phenotypes in AMD. So we decided to just look at our RP mouse models to see if RPE cells were affected and could we save them. So we were able to do that then. We looked at the wild type RPE. This is a flat mount looking at the RPE stained with phalloidin. So we're looking at actin, and you can see a very nice hexagonal regular array. If we look in our retinitis pigmentosa mutant, the RD1, you can see there's quite a bit of morphological kind of problems, and there's actual death. Some of these blank areas are actually missing RPE entirely. And then Don Zach, thank you very much. Don created a very nice promoter for the BEST1 gene, which is specific and high level for the RPE. So we used that to drive NRF2 directly in the RPE in this RD1 mutant. And you can see how nicely it rescues the morphology. And you can even see this at the EM level. So I'm sure, again, many of you know that there are apical microvilli that the RPE cells use to perform various support functions for photoreceptors. Here's a low mag view of the apical surface of the RPE. Here's a high mag view where you can appreciate all of these nice microvilli. This is a wild type. If we look at a mutant, we've lost, again, a lot of the RPE cells. We've lost all of the microvilli. It really looks like a desert. It's pretty awful looking. But if we treat with the NRF2 expressed only in the RPE, again, with the BEST1 promoter, we can rescue that. So it's possible then diseases that hit the RPE and create stress of various sorts may benefit then from an NRF2 treatment. So to summarize what I just told you about NRF2, again, it regulates many genes, actually hundreds of genes, many of which fight oxidative damage. It directly negatively regulates some of the inflammatory pathways. When we look at RNA-seq, as we've done for some of these, there's a lot of genes which change, which have to do with metabolism. We don't know if those are direct or indirect effects of NRF2. If we express it only in the RPE as a hopeful therapy for AMD, we can also rescue at least morphological defects in the RPE. And I showed you originally that if we express it in cones and RPE, I think I forgot to point this out, but our original treatment had a promoter CMV that worked in both. We get better cone survival and better optomotor. So we hope that this can go forward then for some human diseases. And this work was led by Wenjin Zhang initially. She has her own lab now in Hong Kong. And David Wu, who's a physician scientist, ophthalmologist, who's now a faculty member at Mass Eye and Ear. Okay, if we then think about how might we fight inflammation, I mean, we may have already done that with the NRF2. We're not sure where all the benefits of NRF2 come from, but we thought we would look at directly at whether inflammation is leading to death of these cones. And I'm sure that, again, most of you know this, that the microglia are the macrophages of the CNS, including within the retina. And there's now many different functions ascribed to microglia, some of them good, some of them bad, some of them unclear. But they may change their role from good to bad or bad to good, even during the course of a neurodegenerative disease. This is hotly debated, for example, in Alzheimer's and Parkinson's, but there are various roles in wild type animals that have been looked at. Beth Stevens' work has shown that they can eat up synapses, they can participate in these other pathways. So Sean Wang, a medical student when he joined the lab, was interested in whether inflammation played a role. And he started thinking about the role of microglia. So he looked at cross sections of the retina using a GFP strain of mice where GFP was only in the microglia. And if you look at this cross section, there's no green cells here in the outer nuclear layer, but we do see them in the outer plexiform layer, the internuclear layer, and the inner plexiform layer, ganglion cell and fiber layer. If you look in the retinitis pigmentosa strains, after almost all the rods have died, it's hard to see, but there's one layer of cones left here. Now we can see the microglia in the outer nuclear layer, the remaining outer nuclear layer, and Sean quantified that at 2 time points here. He also looked at inflammatory RNAs, particularly cytokines, which often go up in neurodegenerative diseases. And here's just a chart of some of the things he looked at. So blue is wild type, and then the gray bars, as you can see, are upregulated inflammatory cytokine RNAs or complement. So we get a lot of these RNAs going up during the course of the disease. Now, Wei Wang had looked at whether microglia might have a role in rod degeneration and rod removal at the earlier stages in retinitis pigmentosa. And they found that, in fact, the microglia can engulf and lead to the loss of rods in the early stages. And we were wondering if these microglia being in the cone layer might also engulf or otherwise hurt the remaining cones. So we decided to look at that by trying to quiet down the microglia and see if we got more cone survival. So Sean looked at a number of molecules that play a role in the interaction between microglia and neurons, and some of them are shown here. So Sean systematically tested a number of these, and some worked to preserve cone survival and some did not. The way he assayed these, and I'll show you a bit of the data in a moment, but he either drove expression of the gene directly in the cones with the red opsin promoter, or again, he used this nice BES1 promoter to drive the gene expression directly in the RPE. This allowed him to then look at where there might be autonomous effects on cones or non-autonomous effects. And what he found was that fractalkine, this is the molecule that was used by Wei Wang's lab to quiet down the microglia and preserve some rods. We found that fractalkine could also preserve cone survival, but only if we secreted it from the RPE with the BES1 promoter. If we attached it to the surface of cones, and it has 2 forms, I should say, normally, both attached to membrane or secreted. If we used the membrane attached form, it did not preserve the cones. He looked at CD200 and, CD47. This is work we just published. CD47 may be known to a number of you. It is the don't eat me signal that tumor cells put on to protect themselves from death by T cells. So CD47, if we put it on the surface of cones, also protected the cones. And Sean found that SERP alpha is responsible. There's 2 different proteins that interact with it. SERP alpha is the one responsible. Thrombospondin 1 is the other one, and it turned out that was dispensable. He looked at CD200 as a membrane-bound molecule on cones, and that did not work. So I'll just take you through a little bit of the data now. So again, it's the same format. We look at control retinas treated with just a GFP vector. Or we look at those treated with one of our rescue constructs. This is the secreted fractalkine, again, secreted from the RPE. This is the quantification down here in the RD1, so GFP only, and these are some forms that don't work. Again, membrane bound is one of them, but this is a secreted fractalkine. And you can see we get a nice survival over these other treatments. He also looked at the optomotor test, and this is the soluble fractalkine treated, again, acuity over age, and you can see that we can preserve some vision with this treatment. And then we wondered, well, how does it work? So we assayed whether rod survival was improved, and it was not. We asked whether the microglia cell number in the photoreceptor layer was reduced, and it was not. And then we looked at the RNA readouts that I showed you earlier for inflammatory cytokines and complement, and they were not reduced. And then we did an experiment we should have done to begin with. So we should have asked, are microglia important in cone survival? And we can do that by wiping out the microglia with a drug, the PLX3397 drug, which works on a particular receptor on the microglia. And if you treat the animals long enough in their chow with this molecule, then the microglia are dead. And we assayed that by FACS, and in fact, we could get rid of greater than 95% of the microglia with this treatment. So we did that in just GFP treated animals, and we saw with microglia present versus absent, there wasn't a big change. In fact, it was not significant. You can see quite a range in the data here. This is typical of our experiments with these models. But there was no significant improvement in cone survival if we wiped out the microglia. And in fact, those inflammatory cytokine RNAs also went away with this drug treatment. So this says that cones in their sort of their net effect, microglia and their net effect on cones is neutral. Here's the data I already showed you for soluble fractalkine. And then we asked, well, does the fractalkine need microglia for the survival promoting effect? They're the only cell type in the retina that we know of that actually has the known fractalkine receptor. So we treated then the PLX, the microglia depleted animals with the fractalkine, and we got, it looks almost like a bit of an additive effect, but again, not significant, but we didn't lose the effect. So that means microglia are not required for the fractalkine effect on preserving cones. And right now we don't know what cell type or what mechanism is responsible for the fractalkine promoted cone survival. Okay, so just to tell you another anti-inflammatory that Sean used. So TGF-beta 1, 2, and 3 work through a common set of receptors, and TGF-beta 1 is a fairly pleiotropic molecule that can be, again, both good and bad in terms of disease progression. So he treated with these, and he found down here, again, we have cone counts over time, or sorry, at one time, this is P50. TGF-beta 1 gave a very nice promotion of survival of cones. This was expressed from cones, and that's relative to GFP control or beta 2 or beta 3. And we don't know why beta 1 works and beta 2 and beta 3 don't. Again, as far as we know, they use the same receptor, but beta 1 looked good. Like all of our work, I haven't shown you all of it, but we always test in multiple models. So this is RD10 and this rhodopsin null. And again, you can see beta 1 preserves. If we looked at the light dark box test as another way to assay vision, this is very low acuity vision, but we just thought we should use another test. We find that the animals also prefer to be in the dark. That's over here. This is the TGF-beta 1 animal versus GFP, and this is wild type. So again, sighted animals prefer to spend more time on the dark side of the box, suggesting that there is an improvement in at least light perception with TGF-beta 1. Oops. We also wanted to know, again, does microglia play a role here? So we wiped out the microglia again, and this time we lost our effect. So that's over here. I'll just draw your attention to this. So this is TGF-beta 1 in an animal with microglia. We get a preservation of cones relative to control. But if we either wipe out the microglia or we block the known receptor with some drugs, we lose the effect. So this might be more straightforward. The TGF-beta receptor on microglia appears to be necessary for the benefit from TGF-beta 1. So that's kind of interesting, because I told you that microglia in general don't seem to have an effect on cone survival. This tells us then that they can be induced to be supportive of cone survival, and we're trying to track down how does that one work, and we don't know at the moment. So to summarize this part, microglia reside in the photoreceptor layer during the period of cone degeneration, but they themselves do not cause the cone degeneration. Soluble fractalkine, or TGF-beta1, or CD47 on the surface of cones. I didn't show you that data, but it was recently published if you're interested. All of these improve cone survival in all 3 models that we tested. They also improve cone vision by the optomotor or the light dark box test. And it could be then, or from the TGF-beta1 results, that microglia activation rather than quiescence might be therapeutic during cone degeneration. We'd like to understand how. Okay, so that was what I wanted to tell you about inflammation. We then wanted to address metabolic problems, and we've done this over a number of years, starting with Claudio Punzo, as I showed you with the insulin injections. We've been trying to track down what is the most upstream problem that the cones might experience that leads them to lack of mTOR phosphorylation and chaperone-mediated autophagy. And I should say that we have a lot of consistency arguments, but we are not totally we haven't totally proven what our main hypothesis is, which is there's a glucose shortage. So just to review for you, work in the field, Jim Hurley is very responsible for a lot of this. Our lab has done some of this. It's a way of looking at the RPE and photoreceptors as having a symbiotic relationship regarding not just things like outer segment removal, but also nutrition. So the choriocapillaris is a very rich plexus of blood vessels that supplies the oxygen and the energy for photoreceptors via the RPE. So for example, glucose can go through the GLUT1 transporter in the RPE and be delivered to the photoreceptors where glycolysis, aerobic glycolysis, runs at a pretty fast rate. And they also are carrying out OXPHOS at a pretty fast rate. This glucose results in the production of lactate. They have a lot of the LDHA enzyme. And there are 2 small transporters, MCT, 3 carbon transporters, MCT1 and MCT3, that can accommodate the lactate. So as lactate is being pumped out by the photoreceptors, it's taken up into the RPE where it's thought to fuel their metabolism. And any excess can go out the MCT3 transporter on the basolateral side. So the idea then is that rods are providing the fuel for the RPE in normal healthy retinas in exchange for glucose. Now, when the rods die, then there's 95% of the photoreceptors in the mouse are rods. Then obviously there's going to be a huge decrease in lactate production. And this might lead to the RPE holding on to the glucose for its own benefit. So we were thinking about this hypothesis, and we were thinking, well, if the cones can't get enough glucose from the RPE, how might we take care of that deficit? And we thought, could we ask the cones to make their own glucose? So Yash Chinchuri, a postdoc in the lab, took this on. And this is a little bit of a crazy idea, but we thought, okay, could we use the same enzymes that the liver uses to produce glucose and deliver those genes to the cones and see if it helps with their survival? So this is the glycolytic pathway. A lot of the enzymes in this pathway are also used to make glucose in the process of gluconeogenesis. Here's 3 enzymes that are present. Mostly, if you look throughout the body, they're only in the liver. And we looked in the retina, they're not in the retina. And so then we delivered these 3 with 2 different AAVs. And we asked, will the cones survive longer? And amazingly, they did. So this is, again, these GFP counts in the center or the periphery of the retina. For reference, this is the GFP only treated retinas. This is the NRF2. We wanted to compare. We get a nice survival. And these are the 3 gluconeogenic enzymes delivered, and it's both center and periphery. We get a nice boost. And then we thought, well, gluconeogenesis could supply glucose, but there's probably still other problems. Again, oxidative damage, inflammation. So if we put them in together with NRF2, might it be additive? And it was basically additive. So we get a much better survival if we take care of things with NRF2 and we supply the cones with more glucose. So this is actually something we're pretty excited about, but we think there may be problems delivering 4 different genes through AAV to the retina. I'm told by people the FDA doesn't like that. I'm not sure that's true, but right now we're trying to also find simpler ways to deal with these metabolic problems. So Sawyer Shu, when he joined the lab as a postdoc, was thinking about this, and we were thinking, well, okay, it could be glucose supply through the RPE. It could be glucose uptake, something about the cones don't lead to good uptake, or it could be some type of mismanagement, not sure what, within the cones. So Sawyer then started trying a whole series of genes that might affect those processes. For example, he added back the GLUT1 transporter. Wenjin Zhang, by the way, also did that. And he used different promoters. He used the red opsin promoter, 2 different versions, depending on how big the gene was that we wanted to express. Or this is a new cone promoter that was given to us by Botan Raska. He put in a number of different genes, and basically nothing really gave a boost to cone survival except for this one called Txnip. So I'll tell you about his recently published work on Txnip. So Txnip is a multifunctional alpha arrestin protein. So it's not a beta arrestin, as many of you may be familiar with from phototransduction. It's an alpha arrestin protein, which basically is a family of proteins that interact with multiple protein partners. And Txnip is one of those. So one of the things that Txnip is well known for, particularly in the diabetes field, is that it can remove the glucose transporter from the plasma membrane. So it thereby can regulate how much glucose is coming in. It also can bind to thyroidoxin, which is one of these proteins which fights oxidative damage. It also can have effect on fuel sources. A nice study from muscle effects of Txnip shows that it has a role, not clear what mechanism is used, but it has a role in allowing cells to choose different fuel sources, particularly if glucose is not available. So we saw that Txnip could benefit the cones, and then we were wondering, how does it do that? So there were some nice mutations or alleles published in the literature that changed some of the binding partners and functions of Txnip. So we started testing those systematically to see what matters in terms of the Txnip binding partners for this survival. And what Sawyer found was that this one amino acid, cysteine 247, if we changed it to a cysteine, it was published to not then be able to bind to thyroidoxin. And when Sawyer used this relative to the wild type Txnip, this gave a better boost to cone survival. This could be because it's releasing thyroidoxin then to go off and do its antioxidative damage role. It's also freeing Txnip to do whatever else it does, which might benefit cones. So we're not sure which or whether both of those are important, but this was our best rescue allele. Now we asked questions about other possible binding partners and roles, and I'll tell you about some of that. But first, I'll just show you some of the data. So again, here's a GFP treated control. This is the number of cones in the center in this particular model at this time point, and we basically doubled the number of cones with the Txnip C247S allele. We then again measured survival with that allele in 3 different models, RD10, rhodopsin null, as well as RD1. It can benefit all of them. We looked at the vision. Again, here's optomotor on the RD10 and the rhodopsin animal, rhodopsin knockout animal. Again, we could rescue some vision. We then started wondering about the fuels. Again, let's say glucose isn't as available to the cones for whatever reason. Maybe then having the cones directed towards alternative fuels would be beneficial. So we looked at that. There are various genes or enzymes that are rate limiting for the use of alternative fuels. So if lactate is high, the LDHB isoform can drive then the transformation into pyruvate, which can enter the TCA or mitochondria and eventually the TCA cycle. So this was a possibility. Ketone catalysis can also be used as an alternative fuel, and a rate-limiting enzyme is OXCT1. Fatty acids can be used, and lipid transport is a big part of that, and the CPT1A is a lipid transporter. So we tested whether any of these things were necessary for Txnip's benefit to cones. And we did that by supplying, along with our vectors, our AAVs I already introduced, a vector that had the LDHB or OXCT1 or CPT1A shRNAs. So to knock these down, to knock these down in the presence of Txnip and ask, do we wipe out the Txnip benefit if we take away, for example, LDHB? And that turned out to be the case. So if you look over here, here's the control. Here's this is no Txnip. This is Txnip with an irrelevant shRNA. We still get the I'm sorry, my mouse is very sensitive to advancing slides. Anyways, we have the Txnip rescue, still the normal wild type level if we include an irrelevant SI. But if we include the LDHB SI, we lose the Txnip benefit. So LDHB is required for the Txnip benefit to cones. but we did not find that type of dependence upon ketones or fatty acids. So we're thinking then about LDHB and what might that be? Why might that be? It could be that, again, it's leading to more pyruvate. And if a cell has more pyruvate, it may also have healthier mitochondria. So while we were thinking about that, we then went with RNA-seq of Txnip treated cells, and we found not as many changes as we thought we would, but 3 that we got that were significant were 3 of the genes for mitochondrial respiration. So we started looking directly at mitochondria, and these are some EMs. This is a control RD1. These are rather sick-looking mitochondria. If we add Txnip, they're bigger, and the cristae look a lot nicer. If we again add our favorite allele, we get even better. And this is quantified over here in terms of average size and cross-section on these EMs. We looked at something called the JC1 monomer, which, depending upon the health of the mitochondria, it's a monomer form when they're not that healthy. It's an aggregated pink form when they are healthier. So again, you can see more pink to green, and the ratio over here shows that they are healthier. This is basically reflecting membrane voltage. We look with another membrane voltage and also the ATP pump function. So I don't want to go through all these in detail. This is all published, but these assays told us that the mitochondria are bigger and healthier and can perform their functions at a better rate when they have the Txnip present. So then we wanted to know, is the mitochondrial benefit all that you need for the Txnip rescue? Maybe, again, the LDHB is working through mitochondria. So we looked at some of the other protein partners for Txnip, and one of them is this protein called PARP1. Some of you may know PARP1 is pretty prominent in cancer. It's part of the DNA repair pathway. And curiously, if you knock PARP1 out, so now out of a regular wild-type mouse, you take away PARP1, it benefits the mitochondria. So we wondered, is PARP1 as a Txnip partner somehow participating in the Txnip rescue, and could we even just isolate the mitochondrial benefit and ask, is that sufficient for the RD1 rescue? So we got the PARP1 mutant, and we looked at the various assays for mitochondria, and just look at this one. This is, again, cross sections by EM. This is wild type. This is PARP1. You can see bigger and healthier mitochondria if you take away PARP1 in a wild type or now in an RD1 background, and that's quantified over here. So we did get the known PARP1 effect, again, on a wild type background or an RD1. We got bigger, healthier mitochondria, and then we assayed, did we get more cone survival? And we did not. So that tells us that mitochondrial improvements, at least as affected by loss of PARP1, is not sufficient for cone survival. So we're thinking then that there has to be more than one role for Txnip. And I'll summarize that in a moment, but I'll tell you one more thing about Txnip rescue. Again, we're thinking Txnip might benefit metabolism through the mechanisms I just suggested. It might then also benefit in an additive manner if we take care of oxidative damage and inflammation. So I'll just draw your attention to these 2 panels here. So if we put in the Txnip CS allele, we get a certain amount of rescue. And if we combine it with the best one NRF2, again, RPE only, we get even more cone survival, and the cells look even nicer. So this is a cone-opsin stain with just the Txnip rescue, but if we add best one in, we actually get more organized looking outer segments than we get with just Txnip or just NRF2. So we think that we might be getting what we predicted would be some of the benefits. That is, Txnip is giving the cells more possibilities through better metabolism for better anabolism, which might help with maintaining a better outer segment structure. And meanwhile, NRF2 is taking care of some of the inflammatory problems and the oxidative damage. So just to summarize that then, we think that glucose is normally the preferred fuel for cones and rods, and Txnip is removing the GLUT1 transporter from the surface, which, you know, you could have said is going to hurt them because now they're getting even less. But somehow we think Txnip is forcing them to use lactate through the utilization of LDHB. We don't know how that works. We did not see an upregulation of LDHB protein, but we know because of the shRNA data I showed you that LDHB is required for the Txnip-mediated rescue. Pyruvate can certainly then enter into the mitochondria and ultimately the TCA cycle. Because of the part 1 data I showed you, better mitochondrial health is not sufficient, but perhaps in combination with whatever else Txnip is doing, and we're thinking there may be another pathway, but at least there's 2 pathways that we think are part of the Txnip rescue. So again, we're hoping we can use Txnip in combination with other of these rescue genes to get even better cone health. And I think I have 5 minutes left to tell you just one new bit of data that we just published on bioRxiv. And that is something that we've been looking at over the years to ask about far peripheral cone survival. So I don't know if any of, I'd be curious about some of you who work with humans or other species, if you see this as well, but in a wild type mouse, if we stay with cone arrestin, this is a flat mount, we see a nice distribution of cones throughout. But over the years, we keep noticing that the far periphery, it seems to be spared for quite some time. So this is postnatal day 40 in the RD1, but actually we see it in all of our strains. So RD1, RD10, and rhodopsin null. And note, this is more than 1 year of age for the RD1, and these dorsal peripheral, far peripheral cones are still there. Same for the RD10, same for the rho null. And we quantified then these cone survivals. Again, this one's a year and a half or something. It's really very extended. So again, we had been seeing these over the years, and we kept thinking, you know, that's odd. Why are these cones in the far periphery being protected? So we decided to try to figure it out. And Ryoji Yamamoto, a postdoc in the lab, decided to take this on. So he did an RNA-seq of center versus periphery. And we look for peripherally enriched or de-enriched genes. And here's a partial list of the data that he got. And one that jumped out at him was a synthetic enzyme for retinoic acid, ALDH1A1. He's looked at several others, but I'll tell you the story about ALDH1A1. So first he looked to confirm the RNA analysis by doing fluorescent in situ hybridization in the center and the dorsal periphery. This is the ALDH1A1 probe, and you can see we have more signal in the far periphery in the dorsal region than in the center. We then thought, is there actually more signaling from the retinoic acid pathway? So that's an enzyme that makes retinoic acid. And as many of you know, retinoic acid is detected or bound by a receptor, then turning on genes in the genome through what's called the retinoic acid response element. And there's an animal, transgenic, which has that very element driving LAC-Z. So we got the wild type and we looked at the LAC-Z activity, and you can see it's right where we would hope to see it. That is, it's where the 1A1 enzyme is, and it's where the prolonged cone survival is. So at least there is retinoic acid signaling right where the those cones that have prolonged survival reside. We then wanted to know, well, which cell type is reading out the retinoic acid activity and which one is expressing the 1A1? I won't show you this, but 1A1 is expressed in macular glia. That's true from our single cell RNA-seq, from the in situ, actually in both human and mouse. And here's the beta-gal readout from the RARE LAC-Z, and it's in Mueller's, as we can tell from looking also at the GS co-staining. So macular glia express the enzyme, and it looks like it's an autonomous response. Even though retinoic acid can be secreted, the macular glia are the cells that are responding to the synthesis of the retinoic acid, which they themselves do. And somehow that's leading, we think, to cone survival. So we then just wanted to check, is this area of RARE LAC-Z or RA signaling really overlapping with the cones? And so this shows you again that at 2 different ages it does. And then we wanted to know, is it required for the peripheral cone survival? So we made this crazy mouse. This is a 9 allele mouse where we knocked out all the synthetic enzymes for retinoic acid. So 1A1 is the one that we found enriched in the periphery. We saw very little of number 2 or 3. Those are other synthetic enzymes, but just in case they were upregulated, if we knocked out 1A1, we went with the full knockout, or the flox alleles, which we could then get rid of all of these by putting in a Cree that we could turn on with tamoxifen, and we bred it into the RD1 background. So the question here was, do we lose the extended peripheral cone survival if we get rid of the RA synthetic capacity there? And we did. So this is that strain if we don't have the Cree present. And if we do, this is the loss then of the cones. So it looks like this is required for the extended cone survival in the periphery. We then wondered, is it sufficient? And there's a way to stimulate signaling or activation through RAREs by taking the receptor for retinoic acid and fusing it to an activator, a transcription activation domain. So we tested that by electroporating into the Muellers the either BFP as a control or this activated allele. And what we got were more cones surviving in the patches that received that activated allele. And we also got the RARE LAC-Z upregulation. We looked at humans. We got some tissue from center and periphery. This is data from 5 individuals. And the periphery versus the center, you can see in all 5 individuals, there's more of the 1A1 enzyme in the periphery. And again, I'd love to hear from people who work with humans about peripheral versus central vision in RP patients. What we understand is that typically when people talk about peripheral versus central vision in humans, they're not really talking about the far periphery, which is what we're assaying here. If we read some of Anne Malam's data, she says that most of the peripheral loss that initiates RP loss is in the mid periphery, and then it proceeds centrally and peripherally, but it doesn't actually initiate at the far periphery. But I'd love to see more data from humans about that, if anybody could help us with that. Anyway, just to summarize this part then, it looks like in a normal animal, we have a 1A1 synthetic enzyme enriched in the far periphery, particularly dorsally. If we remove all the synthetic capacity for retinoic acid, we lose that excess peripheral survival. And if we instead add signaling through the fusion of the activation domain to RAR, we can then preserve cones even in the ventral areas, even in the more central areas where we don't have the extended cone survival. So to summarize this last part then, these gluconeogenic genes provide a survival benefit for RP cones. We think they might, we did not show this directly. We've tried to do metabolomics. We haven't been able to, but we think that they are allowing the cones to make their own glucose. And at least those same AAVs do stimulate glucose synthesis in tissue culture cells. Txnip blocks glucose uptake, but it forces the cells to use the lactate via LDHB. We're not sure where the lactate comes from. It might be coming from Mueller's. It might be coming from the internal or the retinal vasculature that's more in the central part of the retina. It improves mitochondrial function and structure, the Txnip does. And Txnip provides an additive benefit to NRF2 when we put the NRF2 in the RPE. And the retinoic acid signaling provides peripheral cones with a survival advantage. So what are we doing next? We'd like to test these things in large animal models. We have started to try to do the rescue by later infection or by turning on genes later. Yep. We're looking at combinations and hoping some of those will continue to be more effective. And we'd like to develop this as a therapy, not just for RP, but AMD and perhaps other diseases. And we've been fortunate to have funding from a number of sources. So I'll be glad to stop there and take questions.
Yeah, as expected, outstanding. So if anybody has any question, please drop me a note that you have a question. You don't need to write those questions. Maybe we'll start with the room here. Sam.
Hi, Dr. Cepko. Really nice talk. I have 2 questions about the NRF2 study. First one is, which serotype of AAV did you use? The second one is, did you look at how prolonged the expression is?
Yeah, so we use AAV8 for almost everything we've done. We tested a number of serotypes and we found AAV8 was really good for both RPE and cones. So we've been using that one. And then NRF2 is interesting. So NRF2, many of you know, is regulated at the protein stability and transcriptional level. It's normally degraded through, it's made in almost all cells and then normally degraded when there isn't any of the, for example, oxidative damage or other types of stimuli. So the KEAP1 is a protein that leads to its degradation by proteasomal degradation. So when we put in NRF2, we were wondering, would we overcome the KEAP1 mediated degradation, or how would the levels look? And I can tell you that we can't see the NRF2 protein in our rescue constructs and cones. We can see it in tissue culture cells, no problem. So our construct expresses it, but we can't see the protein. Now, some people might think that's bad. I actually am very happy about that. And the reason is I told you it regulates hundreds of genes. And I was actually worried that we would have some sort of side effects or noxious effects from turning on so many genes at a high level. And in particular, if you put in too much of a transcription factor, there's something called squelching that can happen. Squelching is the idea that you have a transcription factor and it kind of sucks up a lot of the basal transcription machinery, and that can lead to toxicity in cells. And in fact, we've seen that with other transcription factors that we've driven at high level. So the fact that we don't see the protein, I think, is actually good. We clearly get an effect. So I can't tell you about protein levels over time because we can't assay it. Does that
Let's move to Danny.
Thank you, Chris, and thank you. That was a really outstanding talk. It's so much information, novel information to process. My question is specifically about the microglia.
Yeah.
Two parts there. The first part where you're using Plexicon, in both situations, you're using the Plexicon drug. And as you probably know, Plexicon will wipe out not only microglia, but monocyte-derived macrophages that get recruited in. It's also, as you know, a TK inhibitor, so it has off-target effects, one of which would be FLT1, which you showed picked up. So I'm wondering if you put any thought into, I know you made the conclusion that microglia are required or not required, but it seems to me that that might be a bit preliminary.
Yeah,
on the second part, you know, with TGF-beta, that's really interesting because, you know, TGF-beta is actually required to maintain microglial identity. So it kind of makes real good sense that microglia would be required there. And going back to the first part, sorry about being a bit scattered here. Going back to the first part, if you're wiping out both the microglia and the monocyte-derived cells, you know, one might be doing good, as we showed, the microglia are doing good in the context of probably predominantly rod degeneration. Whereas we have some evidence that monocyte and others have monocyte-derived macrophages are doing bad, if I can use the word bad. And so you could be eliminating both sides. So the net effect is that you're not capturing a difference. So I wonder if you had any comments on that.
Yeah. No, your point about the monocytes are good when I did oversimplify. So we tried to actually assay the monocytes with markers by FACS at the same time that we did the microglia. And we got so few, both in treated and untreated, it was hard for us to quantify a difference. So I think we're going to have to do that on sections and try to do a very specific search for monocytes and whether they change with the drug treatment. And I understand the drug does kill them, but much less efficiently than it kills microglia, but we need to count them. And we haven't done that.
So they don't hit monocytes, but as soon as the monocytes become macrophages, they're very susceptible to it. And the issue with flow cytometry, it's not exactly very easy. You have to be very good because a lot of the markers is, you know, the difference is less than 1 decade in terms of the log expression. The differences on some of those markers, it'd be advisable to use a lineage tracing with the CX3CR1 CRE. I think that's the best way to go about it.
Yeah, no, I saw that in your work and I certainly appreciated that. And we just haven't done it. And again, we're trying at least initially just to do sections and histochemistry to see if they're there. And so I can't really address your question at the moment.
Well,
only to say, we use 2 different PLX drugs, but it could be that it's hitting the same population. Both of them are hitting the same populations, and that remains to be determined.
Thank you. And I'm happy to help if there's anywhere where I can help. So thank you.
Yeah. And what was your second question? Sorry, I can't remember now. Oh, yeah. So we agree that microglia may have multiple effects, and all we're asking is the net effect after the PLX treatment, right? And the net effect is that we don't get a benefit or a deficit in the cone survival. So it's kind of curious, and maybe the TGF-beta is working on the positive side. So maybe there is a negative and a positive side, and maybe it's enhancing some type of positive activity. All we know is if we treat with the PLX drug, and again, it could be through monocytes, but if we treat with the PLX drug, we are getting a benefit. If we treat with TGF-beta in the presence of the drug, we lose the benefit. So easiest, most parsimonious explanation is that we are inducing something positive out of that population, but we don't know what it is. And we're trying to track that down with RNA-seq, faxing out microglia, looking plus and minus TGF-beta, plus and minus drugs for blocking the receptor and that sort of thing. And we haven't turned up much yet. Okay. We're still trying.
Okay.
So I can't really tell you how it works.
Sure. Thank
Okay,
you. Thank you very much.
Let's move to Anand.
Okay. Yeah. Hi. As always, so, wonderful talk. I have been following Claudio's beautiful work over the years since
Great.
he was in your lab, and also a lot of other works that have come out. But I'm particularly fascinated by the last part that you put just now in bioRxiv that you were talking about the peripheral cones, far peripheral cone. In human, as far as I know, based on Sam Jacobson's work in many diseases, is the foveal cones that are preserved, macular foveal cones that are preserved very, very late in many types of even LCA when everything else is out. But these cones are dysfunctional, but they survive. Peripheral, I have never, seen any reports so far. Maybe someone else has, better ideas. But I was wondering whether you have considered oxygen availability as a parameter for survival of far peripheral cones. You have less vasculature in that area. So those cones might be sort of somewhat more hypoxic than in the central sort of area where you have much higher availability. And could that be inducing or keeping NRF2 or something else going, some of those factors that keeps those cones alive? You know what I'm saying?
Yeah, sure. And I don't know the answer. I'll tell you, we considered, what's different in the periphery versus the center when we first saw this. And we saw that it was more, the cones in the far periphery in the dorsal region were better preserved than in the anterior, posterior, ventral regions. And so it wasn't just periphery, it was actually the dorsal periphery. Again, we were very happy to see a complete match with the 1A1 enzyme to the dorsal far periphery. But I can't tell you about oxygen. It's a worthwhile hypothesis to try to track that down and really see how much is being delivered in these different places during the course of the degeneration, but we haven't done that. And in terms of humans, and that's why I was asking the question. So we have now looked everywhere we can in the literature. We've asked ophthalmologists and asked, do people actually assay, do ophthalmologists actually assay the far periphery in the human case, even in wild type normal? And usually what I have been told is they don't. So what they call periphery is not the true peripheral, far peripheral retina that we are defining here in the mice. And actually, I don't know if you know Mandeep Singh at Hopkins, some of you must know him. We've been talking with him because he's been trying to track this down, both from the functional standpoint, the imaging standpoint, and even some of the histological samples to find out, is there preserved cones in the far periphery in RP patients? And the little bit of data that's there says yes. But I do think this needs to be done much more systematically. And again, if anybody's been assaying their patients or has tissue, we would love to know, what do you see in the far periphery, particularly dorsally? And
Connie, I think we're going to have two comments on your questions that you posed, one by Arthur Desian and other by our good Dr. Jose.
Okay.
Hi, Connie and Anant. I'll comment on the periphery. We have published our work in about 70 patients with ORF15 mutations in RPGR causing extinct retinitis pigmentosa. And we, at least for the first time in our 30-year history, went really, really far out to about 92 degrees in visual field in terms of evaluating both rod and cone function in these patients. And even though many of these patients were very severely affected, nearly 50% of them retained 50% of these patients retained a far peripheral, nasal far peripheral sliver of cone function, as well as rod function. Rod and cone function in about 40% of the patients, another 10% cone only function, and the remaining, none of the patients with rod only function. this function could be really very amazingly good, including in the general range of normal light sensitivity. And we also looked at the nasal visual field, temporal retina, as far as we could, and to evaluate whether environmental light was affecting this periphery under the hypothesis that the nose and the cheeks would be blocking this part of the retina from light, whereas in the temporal field, the nasal retina, there would not be such a blocking. And we found no obvious difference in the peripheral retention. So it was more circular. And thus, at a preliminary suggestion, we thought it was probably not due to environmental light.
Excellent. I'd love to hear. And you, I should have mentioned, you had a study in dogs, I think, where you looked at the far periphery. Is that right? Or am I mixing that up with another study?
I have to check. I have to check on that.
Yeah.
Off the top of my head.
Okay. Well, thank you. That's very nice to hear.
Jose.
Yeah, so I think the dog study was on the RCD1, and there was an evidence that there is a very peripheral rim of cones, mostly blue cones, that are remaining very late in the process. But although we don't test in many patients the peripheral vision, many patients start with a donut deficit, which is pericentral, and there is a rim of visual field that is retained in many patients for a long time before they lose totally the peripheral vision. So this is mostly cone function because we do mostly photopic visual field. So it's right, but the extreme periphery can be preserved a bit longer. We don't do routinely the 90-degree visual field that Art was mentioning, but in many patients you see this donut deficit, which preserves some peripheral vision at least mid stages of the disease.
Well, that's also good to hear. I should have mentioned too, we looked to see if rods were preserved out there, and we did not see that. So that was one of our first ideas. Oh, maybe just the rods are surviving longer. So were the cones, but they were not. So it truly was a cone-specific prolonged survival. So I'm thrilled to hear that you think some of this is true also in humans.
Yeah, it is.
Okay, let's move to Jennings.
Yes, hello. That was a great talk. And
Sorry, I murdered your name when I said thank you at the beginning, but anyways, thank you.
Oh, no worries. It's all good. So you presented some compelling data identifying targets for treating retinal degeneration in humans using gene therapy. But you mentioned potential hurdles in terms of getting FDA approval for this approach. So I was wondering what your thoughts were on pursuing this multi-targeted gene therapy approach in the clinic versus an approach using small molecule drugs that can target similar pathways, you know, like stabilizing oxidative stress, inflammatory and metabolic processes. And in some cases, you know, these drugs have already been FDA approved for other indications, so could, you know, maybe be repurposed for their context here.
Yeah, so we definitely have thought about the small molecule approach, and it may be a really good way to augment a gene therapy approach. So one could certainly use them in combination. So one of the reasons why we went for gene therapy, as opposed to initially just screening a bunch of drugs, was I particularly like the specificity of it. So I imagine RP patients would be treated over a very long period of time, decades. And I do worry if you're taking a drug that can affect some of these pathways are very important to cells. I mean, metabolism, you know, inflammatory or immune system control, et cetera. So treating with a small molecule that will be affecting the entire body over a period of decades worried me from the standpoint of side effects. So I thought if we could do it with a very specific treatment, and again, having these nice promoters that Don and Jeremy and other people have isolated, having those really specific promoters and the advantage of a local delivery gives you very specific, I hope, expression that if there is any type of untoward side effect type of thing, it's very limited to that time and place. So that's why we went the gene therapy route versus the drug. Drugs that have no side effects that someone could take for decades and might augment anything in the eye that we might do would be a good thing. And I don't know what the future holds along those lines. In terms of treating with a couple different genes, I'm actually heartened by the fact that people who are trying to do various types of gene editing are unable to put the entire gene editing machinery into a single AAV. They're having to split it up. And some of that's looking like it's working well. I know, Chris, you're part of a study with David Liu on at least one of the gene editing approaches, but there are approaches where you're putting in 2 AAVs with either 2 parts of a protein or 2 genes. And if those get approved, I think that's a good precedent for us. But I'm also just thinking that I don't know why the FDA would be so against it. People tell me this, by the way, I'm not an expert, but I'm told by people who do more drug development that having more than one vector with more than one gene is not something they'll be a fan of. But if you can show that either alone, that the additivity really works, perhaps it would get through. So I'm kind of hopeful for that. But it might be that we can't do it, and we might have to use a small molecule to augment something we're doing more specifically with a gene therapy vector.
Connie, thank you very much. This was a wonderful morning on Friday, a crisp, sunny day. And we will be amiss if we wouldn't have a final comments and question from our good colleague from Baltimore. And I give you now, Don Zach.
Chris, anything for that sweater. Connie, as everyone said, great talk. I have two sort of unrelated questions, one of which may be more for the clinical members of the group. But one is, given your beautiful work and others, implicating insulin and glucose, are there enough patients who have diabetes and RP to say if the diabetes has any effect on the progression and the phenotype of the RP? And the other question is your very interesting finding with TGF-beta 1 versus 2 and 3. Did you perform any controls looking at other gene expression and stuff to make sure that the TGF-beta 2 and 3 were actually having biological effects, and it wasn't just some kind of artifact of being inactive, that they actually were acting but not promoting cone survival?
Yeah, so good point. We looked at, I think we looked in 293Ts for expression using ELISAs, and that was it. So it could be that, our vectors were making molecules that were inactive. We did not control for that or test for that. And, it would be an interesting explanation. I mean, I guess what we were doing is screening a lot of proteins or genes, looking for positive results and then just going with them and not spending too much time on the things that didn't work. There's certainly a lot of things we could follow up more, but again, given limited bandwidth, eventually we just went with the ones that worked. I would love to hear what the clinicians have to say about the RP and diabetes. What I have been told, and I saw some literature on, is that the RP trumps the diabetes, that maybe due to high oxygen that ends up in the retina. I don't know why, but would some of the clinicians like to tell us what they think about that?
We don't have a taker. So Connie, thank you very much. I really wanted to invite you to join us a week from now. We have 2 more junior people speaking during this hour. And again, the bunch of fantastic scientists coming soon after. So I hope to see you all, and Don, including you.
Thank you all very much, and I appreciate the help on some of the human data. That's great to know. Really appreciate it.